Welcome back, dear students. Today we are going to discuss other ways of performing tissue culture. And first of all, let's start with cell cloning. By cell cloning, we simply mean you are allowing a single cell that you have got from a plant to proliferate, to divide, to multiply, and produce a large number of individual cells which are very much identical to each other in terms of genetic constitution. And all these cells they form a clone. Okay. There are different ways of getting individual cells, single cells from plants, but you are not required to discuss those methods. Simply, you will say you will get a single cell and allow the single cell to form a large number of cells which are genotypically identical to each other. And these clones are used for a number of purposes. For example, the individual cells may be converted to somatic embryos, and these somatic embryos will form the plants, okay, plantlets. Or in some cases, you will allow these individual cells to form the callus, and this callus may be initiated to form roots and shoots, okay, and you are regenerating the plants. And in some cases, the individual cells can be converted to flower buds, and in some cases, in some plants, you can use these cell cloning technique to produce the secondary metabolites, important secondary metabolites. For example, आपके पास एक plant है, and that plant produces a very important anti-cancer substance. But unfortunately, the number of plants of that type in nature is very much limited. It is endangered, you can say. At that time, what you will do is you will take single cell from that plant and allow this single cell to culture, okay, to divide in culture conditions, to produce a clone, and the clone will give you the same anti-cancer substance that you were getting from the plants, okay, by cutting them. Okay. And now you have used the tissue culture technique to conserve the endangered plant species. Another way of performing tissue culture is you are culturing the anthers. This was started by Goha and Maheshwari in 1964, who cultured anthers of the Datura inox in a culture medium containing kinetin, coconut milk, and grape juice. And to their ultimate surprise, they observed the appearance of numerous embryos, and these embryos were later on confirmed to be haploid, as they have originated from the haploid pollen grains. Okay. Actually, their main intention was to study the physiology of meiosis which takes place in the pollen grains which were present inside the anthers. But to their ultimate surprise, they observed the formation of a large number of embryos which were later on confirmed to be haploid. And these haploid embryos were then cultured in stable culture medium and they developed into haploid plants. Haploid plants are very much important in the plant breeding programs. You can use haploid plants. First of all, they can be diploid. They can be made diploid. You can give them the colchicine treatment to make them diploid, and these homozygous diploid plants are then used to crossbred to create new varieties of plants by hybridization. Okay, so simply you can say you are culturing the anthers to produce a large number of haploid plants which are then used in the plant breeding programs. Here you can see in the image, you have cut down this anther and allowed this to grow in culture medium, and after some time you will see a number of haploid plants are produced, and these haploid plants can then be used to produce the homozygous diploid lines which can be used in plant breeding programs. Ovule culture is other way of performing tissue culture. Here what you do is the fertilized ovules are excised. The ovules are taken out from the ovaries and they are cultured in the culture conditions. And this technique is useful in those species where the embryos abort very much early. Okay. And the culture of embryo is not possible due to difficulty in excision at a very early stage. Okay, so you have to remember that ovule culture is performed in those species where the embryos mature very much early. And one more difficulty is there. You can culture the embryos in those species, but unfortunately the excision, the removal of embryo in these species is very much difficult. So you are taking the whole ovules for culture. Okay, and these embryos that you have rescued from abortion, they can later on be converted to the new plantlets. Another way is ovary culture. In ovary culture, you are taking the whole ovary, you are excising the whole ovary from the flowers, and you culture these ovaries on a stable culture medium. And ovary culture is useful for a number of purposes. For example, you have to do in vitro pollination, in vitro fertilization, and in some cases you can use ovary culture to rescue the embryos. Okay, excuse me. Say for example, you wish that you have two plants belonging to two species, and you want them to cross pollinate, but in nature this is very much difficult. What you will do is you will use tissue culture technology. You will take the ovary out and you will dust the pollen grains of other plant belonging to other species, and you are doing the in vitro pollination. And same is the case with fertilization. In some species the cross fertilization is not possible, and you will use the tissue culture technology to and allow them to fertilize in the test tubes. Okay, and you can also use the ovary culture to develop fruits by excising the ovaries from the flowers and allowing them to grow in the culture medium containing coconut milk, auxin, or any other specific requirement. And finally, the embryo culture. It is a culturing of young embryos in a culture medium to form the seedlings. You are excising the embryos from the ovules, okay, and allowing these embryos to grow in a culture medium so that they reach the mature stage, and finally they get converted to the seedlings. Okay, and embryo culture is useful in orchids, which is a group of plants whose seeds lack any stored food. In orchids, the embryos, they do not reach the mature stage because they lack the stored food in the form of endosperm. And when you are using the embryo culture, you excise the embryo from the seed and allow this embryo to grow in the culture conditions where all the essential nutrient requirements are supplied. Okay, and embryo culture is also useful to escape seed dormancy. In a number of species, the seeds they take some time before they germinate. By using the embryo culture, you can directly allow these you can directly allow these embryos to grow into the new plantlets without dormancy. Okay, this is all about the different ways of having the regeneration of plants, the different ways of regenerating the plants through tissue culture technology. I hope you all understand this. Thank you and stay blessed.